여러분 안녕하십니까? 방금 맞았습니다. 한국말 잘 못해요. 한국에 태어났어요. 그렇지만 한 살에서 부모님은 캐나다 이민 가시고 계속 캐나다 살았어요. 이 정도만. So I'm going to transition into English and let our wonderful interpreters do their great job. So good afternoon, everyone. My name is Kim Han-dol or Han-dol. But I do not play paduk. I am not like Kim Se-dol, but I should be two better. So today I'm going to talk about how to solve small molecule drug discovery with generative AI. And when I say how to, it does not mean that we have already, but this represents a good path to doing so. So first of all, I've been listening to many presentations today, and also I participate in many conferences, and we talk about biotech, and we talk about drug discovery, and we talk about AI. One thing that I would like to emphasize is that in drug discovery, biology and chemistry, although they work closely together, are very different. And the first thing I would say is that the Nobel Prizes for chemistry went to, obviously, as we know, the crew behind isomorphic and AlphaFold, Demis Hassabis and John Jumper, and David Baker, and they're well-deserved. However, are they really chemistry? I don't think so necessarily. We're talking about proteins, and maybe we can argue it's biochemistry. But when we talk about chemistry, we talk about small molecules. We talk about moving, rings and side chains and trying to synthesize small molecules that are therapeutics. We're talking about medicinal chemistry. We're talking about hoods and fumes. And for chemists, especially in drug discovery, the AI revolution has not yet happened. We call it biotech for a reason, because the biology leads. The biology chooses the target, the protein, the enzyme, the gene. And then the biologist turns to the chemist and says, now you have the easy job. Go make me a molecule that will bind selectively, very potently, that is really synthesizable and will not kill the person and has the right therapeutic index. Oh, and also it must be novel. These are very hard things to do. So as a result, I would submit today that biotech and drug discovery and the AI applications, it is a tale of two modalities. In biology, if you're designing antibodies or biologics, of course you're going to use protein large language models. And we have some incredible researchers speaking now, talking about this. One-dimensional string of amino acids, and then using that to predict structure. This has revolutionized the development of biologics. AI for small molecules, not as revolutionary. It's simply the fact that we are solving different problems, and we all talk about AI as if AI is one thing. But as Nahid Kurji, my good friend, said earlier this morning, there are many ways that AI intersects with drug discovery. So really, when we talk about the application of AI in small molecule therapeutic development, which is what I'm going to constrain my discussion today to, what we see are the use, and I saw it in the posters, and some great posters outside, but I also see it at Big Pharma. I see it throughout the industry where chemists are using extremely low-powered, simple machine learning algorithms and not applying the state of the art. And I will explain why. There's a very good reason for that. So today, I'm going to introduce our company very quickly. It is a commercial, so I'm going to be very fast. And I'm also going to talk about AI and a slight history of AI for drug discovery, the difference between generative models and discriminative models, and the fact that, as we all know, chemical space is massive. And this feeds into why older and simple machine learning models, such as support vector machines or random forests and XGBoost, deliver state-of-the-art results, even though these models are 20, 30 years old. Okay? These models were state-of-the-art back when Kim Young-sam was president of Korea. But why do they still work? And I will also talk about data. And everyone talks about data, and yes, in machine learning, you always want more data. But I will tell you that data is not the main problem. And I will submit to you that the use of the algorithm is the bigger problem, because we have to accept there's a limited amount of data that's available. And then I'll talk about how generative AI works, and I'll give you a very simple, high-level analogy. And then we'll talk about the impact of AI for drug discovery on the biopharma industry. And then we'll close for questions. So this is our product. Our product is called Enki. It is an AI platform for drug discovery. The problem that we solve is early stage small molecule drug discovery. And I generated this image using Midjourney, so it's AI generated. But for any team leader of a chemistry team, that's the man on the left, and his team looking. These are very hard problems to solve. Every year, the industry spends tens of billions of dollars in order to try and generate early stage assets. Our company, Variational, has created Enki. It is a foundation model for the rapid generation of small molecule therapeutics. We have Enki Lead Generator, which generates leads that can go directly into lead optimization very quickly. It requires no data from our customers. It is a pre-trained foundation model. We also then have Enki Lead Optimizer that can perform lead optimization. Tell us which scaffolds you want to constrain, and we will do a very effective search of local chemical space to optimize PK, tox, potency, selectivity, etc. And then finally, we have Enki Enterprise, where we work with larger pharmas that have enough data, where we fine-train our foundation model and deliver better value. And as per the introduction, we're working with Merck. We also have worked with Xenon in the space of CNS. We're working with Rakovina, DDR for oncology, and InView in IO. But we are indication agnostic and can be applied to a wide variety of targets. At the end of the day, our goal at Variational is to make the scientists happy. We want to turn this sadness into this. So Jensen Huang, the CEO of NVIDIA, has said, AI will not replace your job, but humans using AI will replace your jobs. These humans have used Enki, and they're very happy. And of course, that makes your patients and clinicians happy. And our goal is to redefine the unit economics of drug discovery. And that is the unmet business need that we solve. So let me go into very, very basic beginnings, because we talk about AI. And for those that know, there are basically two kinds of AI. There's discriminative, and then there's generative. And before 2022, the only AI people had heard of was discriminative, even though we never called it discriminative. This is the AI that was in, your Netflix or your Kakao that would surface, coupons or content recommendations or in e-commerce. And the job of discriminative AI is to classify, right? It's trained on many samples, and it's used in diagnostics. It's used in medical imaging. You train on many samples, and then you give it a new sample, and you say, what is it? And it's very good at predicting. And then in 2022, the whole world went crazy with generative AI, and mainly driven by ChatGPT. And all of a sudden, everyone was talking about generative AI. Before 2022, no one called it generative AI. We called it generative models. And large language models are not the only generative model. There are many. There are flow, there's normalizing flows, there are GANs, there are VAEs, etc., diffusion models. But most people only know about ChatGPT and Claude and various other models. The difference between generative AI and discriminative AI is discriminative AI classifies. Generative AI creates. Generative AI is trained on a whole bunch of data, and it learns what makes something, and then it creates a new thing that it did not exist before. In drug discovery, early on, the first generation of drug discovery companies were using discriminative AI because that was the AI that they knew, and that was the AI that was the most advanced. And first generation AI for drug discovery companies were essentially screening companies. But instead of using, docking scores or using docking packages like, OpenEye or Glide or AutoDock, what they did, instead of creating docking scores, what they did was they just did rapid prediction. And they would assemble very large libraries so that maybe you don't have to do an experimental screen of tens of thousands or hundreds of thousands of molecules. Or maybe you don't have to have a very large high throughput screen and pay Wuxi Aptec, $1 million for many hundreds of thousands, most of which are not good anyway. And maybe they don't have the reagents anymore. So the experimental high throughput screen still being used, but virtual high throughput screening was now screening maybe millions or tens of millions of molecules. Of course, they would do this in silico. And then AI for drug discovery companies came along, and now they're screening billions or tens of billions of molecules, and they can do this very efficiently with good predictions. However, no one trusts docking, no one trusts virtual screening. Just like in a normal screen, you have to do secondary, tertiary, you triage. So all of the great hits that you get, many of them go away. But there's a more important problem with screening, is that it is a small sample of chemical space, and it does not capture the hugeness of chemical space because the space of all possible drug-like molecules is 10 to the 60. It is a universe of possible molecules. Even with virtual docking libraries or AI-based libraries of tens of billions, or even with a trillion make-on-demand library, that is still 0% of chemical space. But it is the best way to do it because if you want to randomly sample chemical space to see if there's an active, that's stupid and it will never work because it's too big. So what do? We are forced to follow what we call the drunkard search or the street lamp problem. We're looking for molecules. We don't know where they are. They could be anywhere. So what do? We look where we know. Right? And what did Sir John Black, the Nobel laureate, said? The best way to find a new drug is to start with an old drug. The best way to start a new hit, to find a good hit, is to start with a known active, right? And then you use your chemistry knowledge and your in silico, power to try and scaffold hop into novelty while maintaining potency and binding, right? This is a very hard thing to do. And the fact is, a lot of this is based on trial and error and experimentation. But the main problem is this. After you have nominated your target and you are in early discovery in the hit ID or hit to lead phase, you are starting at the same points that everyone else starts. Everyone starts at the same points. You go get a screening deck from Wuxi or Charles River or someone else. Everyone's using the same screens and everyone's going after the same targets at the same time. You want to do a DEL library? You're using the same DEL libraries everyone else is, and then you've got to work through all of that. You want to use a virtual high throughput? So what I'm getting at is the starting points are the same. Then it relies on the individual ability and skill and virtuosity of your chemists to find good compounds and leads. And of course, we all know, regardless of how good your chemists are, they need help to de-risk this. Because the fact is that only 50% of programs make it out of lead optimization, right? Of course, if you kept spending money for 20 years, maybe you would get out of lead optimization, but sometimes too much, right? So the pressure is unbearable. And here's the other part, is that when you start with known actives and scaffold hop to freedom to operate, or FTO, while trying to maintain potency, your efforts are constrained by the similarity principle, which is that molecules with similar properties, right, tend to have similar structures, tend to have similar potency. Molecules with dissimilar or far away in chemical space tend not to have similar properties. So this is the fundamental problem of drug discovery, both from a chemistry perspective and also from a machine learning perspective. The most important thing is that when molecules are distant from one another in chemical space, as measured by, let's say, Morgan fingerprints or Tanimoto similarity or Tanimoto distance, it's less probable that they will have the same biological effect. And we see this in the application of various algorithms in drug discovery. So here you can see that it's tracking very simple models, the one nearest neighbor uniform, which is in yellow, KNN, and then random forest on ECFP, which is a workhorse, right? And on the y-axis, you see the mean squared error, and on the x, you see the Tanimoto distance. Going left to right, right meaning more dissimilar. And what you see is the error increases as you get more and more distant, measured by Tanimoto. And there's this concept of applicability domains that exists within SAR, within chemistry, and that once you are out of an applicability domain, then your predictions become less and less accurate. And that constrains the ability of any machine learning model, even the simple ones, to perform extrapolation, to go out of domain and do efficient and accurate prediction. What you're left with is interpolation. And interpolation is great if you're looking at something retrospectively. But if you want to apply it prospectively to find new chemical matter, it's not very good. And of course, in deep learning there, that's us. So as you'll see, the error does not increase as much with an increase in distance based on Tanimoto. And of course, we want extrapolation. That is the promise of AI for drug discovery. Otherwise, why use it? Which explains the reason why it's not broadly used. Because people are using these basic models because they perform very well retrospectively, but they're not finding novel hits. And why would you trust these models to find novel structures while maintaining potency or conserving any other property that you want? And so the sad fact is that if you do a survey of comparative AI approaches, simple AI models are state of the art. I saw XGBoost outside. I saw, random forests. I saw the usual suspects. And of course, at our company, we're machine learning researchers. We're scientists in machine learning. I mean, not me, but my team members. And the fact is it's very frustrating. Why are these very simple models outperforming more complex deep learning models? And perhaps you've had firsthand experience in this, right? And again, I'd like to emphasize it's because the retrospective accuracy does not reflect prospective utility. And so people are using machine learning for drug discovery in small molecules in the way they know how, not in the way that it should be used. And this is one of the reasons why AI has not delivered on the promise that it was having before. And here's my controversial statement, is that the data is not the problem. Many people say, oh, the data is the problem. And my colleague from CAS, talked about the data. And let me be very honest. Yes, great data of high quality, of incredible diversity, and of massive volume would be great. But sadly, that is not possible. The fact is that a model is composed of an algorithm plus data. Okay? And the fact of the matter is the amount of data that exists, experimental data I'm talking about, to train machine learning models is relatively fixed. Every year it grows incrementally. Even if you were to take all of the data that exists in big pharma, you're only talking about maybe 5 times greater than the data that exists in the public domain. At most, maybe 10 times. But that is not enough data to give these pharma companies the edge, and I'll tell you why. And it's because of the algorithm. The conclusion here is that experimental data is so expensive to generate, you cannot do it only for the generation of data. You can only do it when you're looking for drugs. And as a result, it's constrained. The sad fact is that everyone is using the same data. Therefore, the algorithm has to be better. And that's where you get your outsized important performance. In AI, in the rest of the world, when you train a large language model, you may remember GPT-2, GPT-3, GPT-4, 13 trillion tokens, because the training data is free. You scrape the internet, right? And so as a result, large language models can continue to innovate and improve in performance because they have exponentially more data. And there's the neural scaling law that applies, that as you add exponentially more data, the performance of a model improves. But that is impossible in drug discovery and small molecule discovery because the amount of data is fixed. So what do? You have to invest in the algorithm, right? We can all complain about data, which we all should, but it's something that no one can fix. So we need to make the best of it. So we see on all measures of performance that when you add exponentially more data, 10 times more, 100 times more, 1,000 times more, 1,000,000 times more, performance increases. And that is using a large language model, for example, or diffusion models. Okay? But that doesn't exist. So what are we left with? What we're left with is the efficient use of generative models. And this is how we do it. And this is obviously an analogy. But you're familiar with diffusion models such as potentially Midjourney or Stable Diffusion or DALL-E 2, where you create a prompt. You describe the image that you want. And every single word in that prompt there's training data to back it up. The model then can create a new sample and a new picture that has all of those properties in it. And these are actual examples from Midjourney 6.1. So smiling girl with short black hair riding a red bicycle over a bridge. It generates novel images that did not exist before. These are not from Google. It just made it. It took me about a minute. You'll notice that in each case, all the properties are met, right? You describe what you want and the foundation model creates it. Well, we do exactly the same thing, but not for images. We do it for molecules. Our prompts are your preclinical TPP. We've trained on over 400 million samples of data and therefore are able to rapidly generate novel compounds that meet all of the properties that you're looking for. And we do it very quickly and inherently solve multi-parameter optimization. Experimentally, we have a 56% hit rate of compounds that are below 1 micromolar. And as you can see, a significant number are in the single-digit nanomolar potency range. And also, these are all novel compounds, not from make on demand. We have 90% rate of synthetic success, and our performance is 10 to 100 times better than competing platforms. What's the impact? The impact is this: better molecules, much faster, and way more economical. And that is what we deliver on a platform basis to our customers. We published papers that show that 100 molecules generated by Enki are worth more than a million molecule high-throughput screen. And I'll end on this. If we extrapolate the impact of AI for drug discovery, specifically generative AI for drug discovery. And Nahid also referenced this, but this is from the Jayatunga paper in 2021, where 24 AI for drug discovery companies in 2021 generated 158 assets, preclinical assets, versus 333 by the top 20 big pharma. There's a follow-up paper in 2023, they tracked it, where the same 24 AI for drug discovery companies have generated over 400 preclinical assets, which is now more than all of top 20 pharma. If we look forward in 5 years, in 10 years, and if this trend continues to hold, the majority of compounds that are preclinical will be generated by AI, not just by these 24 AI for drug discovery companies, but by pharma, by biotechs that are using AI. We want to be the primary delivery of that service. We would like to be the first stage where any company comes to help find these compounds. Because if you are not using AI to discover your preclinical assets, very quickly you'll be outcompeted. It's like a virus. So you need to be ready and you need to adopt. And it could be that in 10 years that the majority of clinical assets are also AI generated. But of course, we still have a ways to go on that. And that is my presentation. Thank you very much.